And then he asked me the end question, do you know who won the Nobel Prize for chemistry for the structure of ribosomes? I will go to have dinner with one of them today and asking if it is actually worth to put some money in your company. You already are in trouble with science and experiments, and if you have also the money on top, the money issue on top is getting a little bit harder. But basically the money that I invested at the beginning for my company, I just spent it immediately, 10,000 euros. I put everything all in.
Amazing. Amazing.
I was paying £1,000 for one room. That basically my fellowship was coming into my bank account and was going out to pay the rent. So that was the financial constraint.
What are the biggest opportunities and how do you see the future outlook?
The rules is basically,
in one word, how would you describe the future of biotechnology?
Combination now of AI and biology.
There are a lot of students watching this podcast. What would be your advice to them based on your learnings?
I never had plan B.
So no plan B.
I always try to do things that I like.
Many things you can plan, many things are accidental.
Either you fail fast or you grow fast.
This episode of the BCI Podcast is powered by Immagina Biotechnology.
Welcome to Biology Career Insights. In this episode, our guest today is Dr. Massimiliano Clamer, a scientist turned entrepreneur with a remarkable journey at the intersection of biotechnology and innovation. Massimiliano is the founder and CEO of Immagina Biotechnology, a company he has been shaping for over a decade and now leading towards new horizons from ideas to IPO. With a PhD in biomolecular science and a strong background in pharmaceutical sciences, Dr. Clamer trained between University of Oxford, the University of Padua, and the University of Trento, building a foundation that bridges world-class research with real-world applications. Today, he continues to drive biotechnology forward as both founder and CEO and chairperson of Immagina Biotechnology based in Trento. We are excited to dive into his story of science, entrepreneurship, and the future of biotech innovation. Welcome to the BCI Podcast, Max.
Thank you, Manish, and thanks a lot for the kind introduction.
Amazing. So, we'll just revisit your childhood for the moment. Tell us from the beginning, how did you kind of end up taking biology?
I was very a kid with a lot of curiosity. So I grew up in a farm with my grandpa and grandma for the first 6 years. I had to speak a lot with animals, and that's probably shaped a little bit my empathy and the curiosity for nature. After school, I went to the University of Padua to study pharmaceutical science. That was a 5 years program. During the last year, actually, I took a very long internship in Germany, in Bonn, to study biomolecular science related to plants. And that was actually very interesting for me. I learned a lot of molecular biology because during the pharmaceutical science course, you do a lot of, chemistry, organic chemistry, inorganic chemistry, the path of drug, you study all the path of drug discovery, a lot of structure. Actually, you don't learn too much about biotech. I went to Germany to Horizon program, was Erasmus Horizon program, and I really enjoyed that period, and I learned a lot about plants and bacteria, and that shaped a lot, brainstorming of curiosity and new things, new people. I went in touch with the academic environment, super smart people, and then when I went back, I got the imprinting of science within me. And then actually after the degree in pharmaceutical science in Padua, I worked in a pharmacy shop for a year. I need money, so I went immediately on the job market, but I fully realized immediately that was not actually my ambition. It was quite a very boring job in a pharmacy shop selling drugs, most of the time to very old people, luckily, but or which you can have very beautiful discussion about life, but not very stimulating on the other side. So I went, I tried to apply for whatever position in academia, and I got a fellowship at the University of Trento, pre-doc, and then I started a PhD on the host pathogen interaction and what happened on the translational level on the gene expression translational level when human cells are infected with the bacterial toxin. Very cool project. And yeah, my supervisor and my tutor were happy with my work, and so they decided to keep me for the PhD. Started PhD at University of Trento. Unfortunately, it was a fellowship that had to be renewed in the middle of the PhD, and they ran out of money, the university. So I had to find my own fellowship to carry on the PhD, and you do a PhD, you already are in trouble with science and experiments. And if you have also the money on top, the money issue on top is getting a little bit harder. But that was actually a very good opportunity because I applied for a fellowship from the economic department to go abroad, Italy, and work in a lab to validate a business idea. So more applicative science. Well, basically, I wrote a proposal, and with my proposal in my hands, I went in Oxford to the lab, Higgin Bailey lab, the founder of Oxford Nanopore, and he was so kind to accept me in his lab and to do science for other 2 years. Actually, he paid me for a little bit at the end of the PhD. And that period was probably best period of my life in terms of, connection, network, science, visibility of science. So how much science around you can touch and you can breathe, right? You breathe science. And and that's it. And that's how I build up my scientific background, because in Oxford, I work on the single molecule RNA sensing. So I start with pharmaceutical background, then a lot of molecular biology, cool stuff in bacterial, plants, and human. And then I went up in doing technology development, RNA sensing.
Fantastic, Max. That is a journey of some kind. But I just wanted to go very back. Like, as a child, did you ever imagine that you would study biology or entrepreneurship later? Like, did you? Because, we all as a child, we dream of a career. So, where you had some fascinating dreams as a child. What was your dream?
Fascinated by science and technology development. It was a fiction in Italy at that time. MacGyver, like a guy, creating stuff, and then whatever. So I was fascinated about be able to create something with your hands, starting with technology base. Starting with ideas, starting with a small concept, and build something that has a utility for whatever family, friend, or maybe someone else. And I was really fascinated about nature. So the contact with nature that I had at early stage in my life. I also living in a beautiful place that is North Italy, Trentino, where you have high mountains, you have big lakes. You're not so far away from the sea. Venice.
So we also have vineyards.
We have a lot of good wine.
Exactly.
We have a very good white wine. The red wine probably is better in Tuscany. The red wine is better in Tuscany, but still, I enjoy it.
Fantastic. No, I always ask this, to all the scientists and founders, whether whether their childhood dream was to become like an entrepreneur and scientist. And in most cases, I realized that no, they wanted to become actor or a musician or something else. And then some teacher in school or in college or during PhD kind of really made them so curious that they just went on to do, science or founding a company. And in your case,
more like, I don't know, quite a natural. I don't know. I didn't plan anything.
You didn't plan.
I didn't plan anything at the beginning. If you ask me what I wanted to do when I was 15, I was say a physician, doctor. Yeah, you like science and working with But then what actually shaped, I had very clear in my mind what I want to do. So entrepreneur on life science from ideas to the market was my period in Oxford, where my mentor there, Higgin Bailey, I was basically absorbing a lot of, the way to approach to science and how he see potential for, transition from the bench to the application area, and also the opportunity to stay in touch with a lot of people working for startups, right? And that was really the trigger that in my mind, had a on trigger saying, okay, you can translate what you're doing in the bench 10 hours per day is something very useful. Coming back to, the 10 years old child, they were looking at the MacGyver fiction. and the TV, right? And so translating something from the table of your mom into something that is functional. So that's probably what triggers me at the during my early stage career. I will always say thanks to people that allow me to do the PhD first, because without that it was not possible. The environment and the lab in Oxford that give me the right direction.
So was it any of, because most people pursue PhD to be in an academic, right? Like become a group leader, professor, that kind of thought occurs when you enter the PhD. But was it clear for you in the beginning that, the route that you chose, was it just because of accidental encounter with the founder of Nanopore in Oxford, or was it intentional?
No, it was not intentional. I think that the only driver for me was do something different and try to shape your life. Having the driver in your hands, doing something that you like. I always try to do things that I like.
That's the big thing.
I like if I don't like something, I basically I don't do it. And this is why I skipped the work in the pharmacy shop. I went to university, and this is when I was in trouble during my PhD. I didn't stay there waiting for someone giving me a fellowship or some money to carry on, right? And I applied for kind of fellowship that were in the direction that I want to do. So I'm doing something applicative. So I applied for fellowship in the economic department, and then I identified the labs. There are many opportunities. The world is so big, right? Identify labs that were somehow linked to with my background, but also giving me the opportunity to move a step forward. So if you have to plan a PhD, I would say plan in a way that will makes you happy, or at least potentially will make you happy in terms of new knowledge and new experience, new environment. Of course, there is always a risk, right? But there's also always a risk, even if I'm sit here in front of the camera speaking with you.
No, I get that risk part, but so many things you can plan, Max. And as you said, many things are accidental, so there's also a component of luck. Like, you landed in a lab that actually clicked with your goals and your interests. So that way, I think you are quite fortunate. But tell me about the challenges, because, PhD is a very challenging journey. Money is one thing. And, in retrospect, you are smiling about it. But I can imagine, after 1 year of PhD in Padua, your grant is not there. You don't have any economic background. You have to write a grant to get successful in the economic department. So, now it looks easy. But tell us, what was going in your mind and how did you cope with it?
I'm a kind of person that is getting quite nervous when there is an accident that is stepping in between me and the target, right? I'm getting quite nervous and try to really think every minute of the day to how I can solve this issue. During a PhD, there are always many, many different challenges. This can be, on the bench, the experiment doesn't work, and this is happening 90% of the cases. Money issue, this is usually during a PhD, you don't have too much issue with money because it's already planned, but in my case, it was different. You may also have an issue that is hard maybe for a young person to enter into a lab, onto an organization that has already some dynamic, some postdoc, or maybe people that are becoming professor that are already established, that are already some dynamic. And usually young people at the beginning of the PhD are very enthusiastic and they want to learn a lot in the short time in order to get published a lot of papers soon. But this is not how it works. So I think that what I learn. So I think that I'm not the right example because I was very, very nervous and I didn't sleep the night and I'm trying to solve the issue.
No, I think you are the right example. You are the right example, Max, because I think most people can relate to it. Because, if you're not nervous and if you're all the time very happy during PhD, then perhaps there's something wrong.
I'm usually a quite happy person, smiling, but I would say that during my PhD, I think that I remember the few days when I was really, really happy. And that was a few days. That usually is one work. It was a lot of hard work and carry on. I mean, I was happy to the environment. Both in Trento as well as in Oxford. I had around very good people to be very supportive. So at the beginning of the PhD, usually you want to rush, right? And I mean, just this is my experience. I was very nervous about the experiments. So I had a timeline in my mind. So I want to do everything according to my timeline. But the plan usually are not reflecting science. I was nervous about the timing, the results. There is nothing that you can do. I mean, you can just be as best as possible in accepting the results, the nature that you have in front of you, and try to, I mean, change the story, adapt your experimental setup, maybe sometimes your goal, depending on what comes out. And this is actually true in life as well, right? I think that probably the best lesson that a PhD can give you is resilience, right?
Absolutely. Totally agreed. So Max, coming back, so you said your PhD was funded by economic department. So what was it all about? Can you emphasize on the topic?
Yeah. It's so in part of my PhD. So part was financed by the biotech department, part from the economic department, part from the University of Oxford. So I have a full mixture. But the one from the economic department was a fellowship for 2 years called Claudio De Mattei Fellowship. It's still a fellowship that is still active. So every year they give this grant, and they pay you a certain amount of money. So you are fully paid, and you can decide to go whatever lab you want to finalize a project that must have an applicative, an applicative context. Can be both in biotech, can be in sociology, can be in economy, can be from many different. It's very multidisciplinary.
And it's funded by Italian government, right?
No, is funded by a private association. There are basically healthy people that every year they give money to this foundation. There's that says
Full idea.
the organization is affiliated with the university. Yeah, there is a board screening for the applicants, the ideas. Then they have an interview. I mean, I would say is an opportunity to exploit the freedom to decide what you do in your life, right?
Yeah, because most of the time PhDs, inherit a project. They go and they have to work in a lab that they got a position. But in your case, you had a grant and you could choose a lab that is of your interest, right? So that way, I think it was a good
you can match of like a kind of lab rotation, but instead then in a specific department is worldwide.
So yeah, coming back to the topic. So what was the topic like? And because I wanted to see, how the foundation behind Immagina built from there. So tell us more about the topic of your PhD.
The PhD was, again, host pathogen interaction, one part of the PhD. So how bacterial toxin are jacking the gene expression of the host cells. In many diseases, you have a mixture of human cells with bacterial cells. Think about the intestine, also some cancer, where you have infiltration, you have infection over the mass, the mass of the tumor. And this bacteria can change the gene expression of the human
Host,
right, of the host. And this can be done with many different tools by the bacteria. One of these tools can be bacterial toxin, and I was involved in studying the effect of one specific bacterial toxin that is a pore-forming toxin called alpha-lysine. So it's a toxin that is a monomer, but when it binds the membrane of the cells, it oligomerize, generate holes, and you punch the cells with these holes, and the cells are usually dying by osmotic stress. But what happens? Sublytic concentration. So when you have a lot of toxin, the cell die. Yeah, a lot of holes, right? But when you use very little amount of toxin, so you have a sublytic concentration, nobody knew what was happening, and we published a paper where, very low concentration of this toxin can reshape gene expression at the level of translation or the level of the translatome. So not the level of the DNA, not the level of RNA per se, but the level that is basically the step before the final proteome, right? And then funny enough, and that was the link between one project and the other. The same protein, the same toxin, was used in Oxford to form a pore in a synthetic bilayer, and they use it as a sensor for RNA strand passing from chamber A to chamber B through the pore. Right? You have a bilayer, you have a lipid bilayer that are divided into different chambers, and then you put a voltage between the two different chambers. You can force a negative nucleic acid to pass from this chamber to the other, and the only passage is the tiny pore, right? And so at this pore was basically the same toxin as before. So that actually was the connection between the first and the second project. Yeah.
Amazing. No, amazing. And, because this itself is so interesting. So didn't you think that, hey, I should continue doing research? When was it that, you thought of enough of research? I need to do something that kind of makes money, for example, or I want to do something which you have dreamt of like a teenager, that let's build a company. How did that transition happen?
That was in Oxford, as I said. And I think it was a mixture of financial constraint. I mean, this is a trigger. Yeah, that was for sure a trigger, because I was paying, I can tell you, I mean, that was a very long time ago, but I was paying £1,000 for one room, and there was me and my wife in a share flat, right? And that basically my fellowship was coming into my bank account and was going out to pay the rent. So that was the financial constraint. At that time, I was not, and that was in my mind mainly, I mean, thinking about, after the PhD, right? But it was not really the main trigger. The main trigger was the people around me. So I was lucky because I was basically going to Egan's lab when the first wave of postdoc and PhD student, ending the PhD, were moving to Oxford Nanopore. And the Oxford Nanopore was already out from the lab. So I was really touching with my hands the excitement to take something from the lab that was workish, and put into a company for a product that will change the way, to sequence genome for everyone, everywhere, right? How they say that.
But Max, you could have an easy way out, like most of us who could just find a job in industry, right? Because maybe that's the easy step forward. Because building a company is another Herculean task, and that too after PhD, because PhD gives you enough stress. So how courageous you were, or how did you gather that courage in spite of the financial constraints?
I think that I was inspired. I'm observing a lot others, and at that time I was inspired, and I'm learning a lot, especially people that are not exactly doing the same job that I'm doing, right, or the same position that I'm doing. I'm learning a lot from other perspective, and I learn a lot from people move to the Oxford Nanopore. I learn a lot from people that were founding other companies in Oxford. Now they changed a little bit, but was this Oxford University Club just behind the Chemistry Research Institute in Oxford, and every evening after work, a lot of people were having drinks and speaking about science. That was great. I mean, after work, everyone was speaking about science, and a lot of people were thinking about to set up their own company, get in touch with VC, and for my first time, for me, listening about people starting something by their own. Third, I was attending business courses at the Said Business School. I was using my PhD hat, easy PhD hat, to get through even for free,
because normally it can be very expensive, MBA from
right? But
exactly.
If you're a PhD from Oxford, right?
Yeah.
I was a visiting PhD in Oxford, but anyway, I was able to attend to many courses at the Said Business School, even for those that I was not getting any certificate. I mean, I didn't care. I mean, for me, it was just getting as much as knowledge as possible, because I was feeling I was really lacking the knowledge of, what is translational science, right? I knew what was science, and I learned it a lot from different universities, Italy, Germany, UK. but I didn't know anything about translational science. So I wanted to really learn it by myself. This is usually the way out. And then I was inspired by a PhD from another PhD student from Ege's lab that he quit the PhD, I guess. I don't remember, but it's still, I need to ask him, but I think that he never finished. And he started a company in Oxford, but he was so enthusiastic about this project. I actually invited him to Italy to present his PhD to my local university here. It was the other half of the PhD. And I'm still in touch with him. And I think that he gave me kind of the final push to, everyone's can do it. You just need a lot of, brave enough and a start. And then you will see if this actually will generate something or not. And he's now actually is CEO of Immagina. It's a company that went public 2 years ago. So he went to US then and started another company. So if Dan will listen about this in the future, by the way, I want to say
No, but I have similar friends who actually went in the US because the risk capital is much more available in US. And that also it's a very entrepreneurial country, right? So, but yeah, it's always about the founder behind, right? So I think
that you can do business in Europe as well as in US. It's just that in Europe, maybe you mitigate a little bit risk, but you have very slow curve. In US, it's a little bit high risky, but everyone is used about the high risk, and the slope is much go up steeper, right? So either you fail fast or you grow fast. That is a point of view.
No, this is such a fascinating story, Max. Coming back, so one question that automatically comes up, right? Because you need capital to start a company. So for the audience, are you from a rich family? Are you from a middle class family? How did you raise the first capital?
Well, I should ask this to you. You're more expert than me in raising money. No, in my case, we're really self-made, so I'm not coming from a rich family and middle class. My mom was working in a hospital as a nurse. My father was working for an insurance company, so salesperson. Basically, I didn't have too much money from myself. Basically, the money that I invested at the beginning for my company, I just spent it immediately, 10,000 euros. Everything that I had on my bank account. Yeah.
So you put everything that you have saved so far into this company, but that is a huge
all in. I put everything all in.
Wow, Max, but
that was not a lot. That's what I don't.
This was not much.
You have $1 million and you spend $1 million.
No. The other day I was talking to the founder of CureVac, and he mentioned, he got the money like 100K, and then and without a contract. So what happened is that after he had spent all the money, the investor actually asked his money back, and then he had to take a lawyer and everything, and then finally the result was that he had to return back the money. Imagine his crisis.
This is even worse. Yeah.
So he had to return the money, and then he was begging to the bank and everything. So in that case, I think you were still okay. This is my money. I don't have to but 10K is not enough in the beginning. So how did the funds come to you?
At the beginning it was 10K from my side, and then there was basically full owner, right? Limited liability company, one shareholder. And only shareholder. But I found the money from the local innovators. After Oxford, I went back to Italy, to Trento, and I went back because I won a small grant to validate a business idea. When I was in Oxford, I saw the opportunity to write a grant, to write a proposal, to get some money to validate a business idea. And in Oxford, I was immersed in a lot of different ideas, and I came out with a simple possibility. Is it possible to purify ribosomes, this huge macromolecular machine, with a small antibiotic? Because antibiotics are drugs that are known for decades to kill bacteria because they bind ribosomes, right?
Mm-hmm.
And they're I said, why don't try to change a little bit the antibiotics in order to use it as
a hook to isolate it, basically.
isolate ribosome, because the only way to isolate ribosome at that time was still now was an old method from 1970 based on a sucrose gradient. So
They do this sedimentation gradient, like you centrifuge it.
and a lot of you need a big centrifuge that is cost €100,000. You need a lot of time. You need to manually prepare this sucrose gradient. Now people automatize it a little bit, but still is a lot of work. So I said, why don't try to do it faster, cheaper, and even better? Try to capture selective population ribosomes, those that are more active than others, right? So I wrote this small proposal with 2 pages, and the local innovation agency said, if you come here and you demonstrate that you can do something, we'll give you $50K. So that came here, I set up the company, $50K, then from my side, 6, and that was enough to work in local incubator that also was part of the university, part was private, and rent a space. And then within a year, filed the first patent on this idea. And that actually, we test many different things. I was me, and I invited actually, with that money, I also paid for 4 months a friend of mine from Oxford, a very good organic chemistry, that was living in my house together with his wife, and for 3 months he did organic chemistry for my company. And
Wow. I mean,
his home with it.
wait a second, Max. I mean, this needs a lot of convincing, because somebody moving with his wife, he was to believe in your company. How did you convince him? And because you couldn't pay him basically a lot, right? So
No, I was not paying him a lot. I think that I still need, probably I need to offer some a good dinner still. But no, I think it was just French. I'm still a good friends with him, and we're very good friends. And it's sometimes when I go to US or Canada, I'm calling him. I think it was just friendship, friendship. We really enjoy stay together. In Oxford, we were in the same office. He was working for another PI. We was in the same office. We were going out for dinner together. Yeah, and we enjoy to stay. I mean, to stay together. And he was ending the postdoc, right? So he had like a period, a vacant period. So he didn't know what to do exactly. And he's like, okay, let's try to do it with you. And in Italy, at the end, was not so bad.
all right. So was this productive enough? Like these 3 months?
Oh, yeah.
Because the money,
worked-
yes.
We worked from 8 a.m. to 8 p.m. every day. Yeah. And
You still are
working, but from the lab, quite happy. That's actually makes the difference, right? The people that you have around that trust what you're
Absolutely.
doing, and mutual trust, I would say. Mutual trust. You do it because you really enjoy to do it, and together you think that if you make it happen, will be something that will develop, right? Something that will generate something else. So is this I would say short-term vision, but it gives a lot of energy.
So the first name itself was Immagina. So this was your imagination right from the beginning.
Yeah,
Okay,
but
because
actually not coming from me, it was coming from an idea from a postdoc from the University of Trento that suggested this name, and I like it.
No, I like it too. Yeah, that's
It's amazing.
okay. So
It's amazing.
yeah.
Because at the beginning it was imaging everything.
Exactly. And in a way, it is among the very few companies that does a lot of translatomics work, right? Like it's an RNA company. So of course, when you are establishing something new in the market, you also kind of, you have the fear because you have nowhere to kind of copy, paste, improve. So you have to build everything from scratch. Tell us about that journey. So what was the next step after this 60K? How did you take the next step?
Now, after this money, this money we burn it out quite quickly. Then I was lucky that I had the support from the local innovation community to pay put a little bit of money, like grant money, to carry on for a while. But soon after, I was lucky enough. So I was flying to US. I remember it was before Christmas. I was flying to US because I said there is no way I can raise money here. They give you money if you want to start a new vineyard, but at that time, now the situation changed quite a lot.
Change.
Now the situation after 10 more than 10 years or 13 years. The situation changed quite a lot, but 13 years ago was not like that. So I was flying to US and actually I fly there. And I was in San Francisco, Boston, and New York, spending basically everything. But before going to US, I had an invitation to pitch the company to a local network of business angels.
In Italy.
And yeah, here in Trento. So
In Toronto.
because I went to the bank asking money to the bank to carry on, and the bank said no way, we don't put money on this. The bank did, but the guy at the bank told me there is another person in the other room that is used to invest in small projects, startups, companies, startups with business angel. So I went to the other room and I had my elevator pitch. But actually he said that he knew me because he was one of the person on the board that evaluated the grant proposal that I wrote when I came back to Italy. And then he said, try to pitch your ideas to in front of a network of investors. So I had my pitch in front of this 20 local business angels. And honestly, I came out from that room saying they would never put the single cent on this. But I was wrong. Actually, I got a call a week after saying that 4 of them were willing to put some money.
So Max, you said you went to the bank and the bank gave you the connection to the angel, right? So in a way, this was really good that you went to the bank, isn't it? Because otherwise you know what.
In that way, yes.
Yes. And the angel in the beginning, he said that perhaps he wasn't interested. And then you got a call from him that he wants to invest, right? This is what the context is.
Yeah, exactly. So the lesson from the bank is try everything you can do because the opportunity may come from
Anywhere.
few places where you never know. Yeah, the business angel there said that if I want to pitch the company in front of a community of local business angel, and I did it. And when I finished my pitch, I was feeling that nothing comes out. I was feeling that nobody was investing any cent on that because most of them were not really involved in the life science sector. But actually it was wrong because 5 days later I received a call saying that they were interested in evaluating the opportunity to invest in Immagina. Thanks to them, thanks this 4 local business angel, we went in touch after a year with another network of business angel that are connected with Austria, Switzerland, Germany, UK. And thanks to them, I actually got in touch and I had the opportunity to pitch the company in front of one of the most famous business angel in UK. Herman Hauser is famous because he built up Solexa, then sold to Illumina.
Wow.
ATM Computing, sold to SoftBank. It's still active.
So
Yeah.
quite a reputed guy.
Yeah. And I remember like yesterday, I went to his office and is actually founder of a VC firm called Amadeus Capital in UK. And the office are close to Buckingham Palace. They went there. And I was very, very nervous, I remember, because, I look at the profile, right? And I pitched the company, and he let me speak for almost an hour, say almost nothing. And then he asked me at the end, right, question, do you know who won the Nobel Prize for the chemistry, for the structure of ribosomes?
Wow.
Yes, or 2 names, blah, blah. Well, I will go to have dinner with one of them today and asking if it is actually worth to put some money in your company.
And was it Venki Ramakrishnan? Because he was in UK, right? Cambridge. Was he the same guy?
Yeah, exactly.
And you joined or he had the dinner?
No.
Okay, he went.
I was out of my role.
Wow. Yeah, that time it was a big boom on this. So I think you were at the right time at the right place. This is also, I would say.
Yeah, I would say that 5 years before COVID. And I think that a lot of information about the ribosomes were already there thanks to this great people that solved the structure of ribosomes, but a lot of also biochemical information, biophysical information about ribosomes, how ribosomes are translating mRNA, the biophysical properties, the single molecule biophysics from many labs, both in Europe as well as in the US, revealing how ribosomes are working, not only how ribosomes are made, but also the dynamic of ribosomes. So there were a lot of information, right? But still a really very basic research, and nobody or very few people were thinking to exploit this huge knowledge and try to find something applicative. For example, now you have companies using tRNA as a drugs, right? To use a tRNA that allows the ribosome, for example, to bypass a premature stop codon, so you can treat all the diseases that have premature termination of translation, so truncated proteins. So I mean, I would say in the last 5 years, there was a boom in the translational side. From translation, I mean, it's not only ribosomes, but everything that is proteins to applicative field, both in terms of biomarker, biomarker for minimal residual disease detection or diagnostic biomarker, then drug discovery, drugs made of tRNA, mRNA drugs after COVID, circular RNA drugs. Yeah, we had the boom of also novel targets. So the proteome is actually expanding because people figure out that ribosome are translating not only the canonical open reading frames, not only the proteome that we know by standard mass spec, but potentially we could have 3 times more protein than what we think. That means much more targets for small molecule, right, or antibody. There's all the field of new antigen, the dark proteome. It's something that exploded last year.
No, fantastic. Because, I see a lot of proteomics company also became, so big in the meanwhile, in the same phase. For example, Olink, and that got acquired by Thermo Fisher, for multi-billion-dollar deal, So I think the field is still having a lot of potential, right? Because a lot is happening. Again, so we'll highlight the technology that, Immagina does. But just to finish the story, so you met this investor. Did he invest or not?
Yeah. At the end, he invested through a family office.
Fantastic.
We had the three round of business angel, let's say business angel, very big business angel, right? In total, were 3 rounds. Then during COVID, we rejected an offer of acquisition, and because one of the shareholders decided to, keep the company European. Company wants to buy us from US, and one of the investor from the small investor, the shareholder, decided why not to keep the company in Europe and grow it up here and create value here, right? And he made a very good offer to keep the team, to build up our own infrastructure. He put money to create the labs, so we have fully equipped labs for everything that we need to do now in the future, and expand the teams 4 times. And we are still growing here now.
Wow!
So that was the good choice in terms of, keeping the value and growing the technological value in Europe, but also the human value in Europe as well.
All right. So everything started with the idea that was basically to isolate the ribosome with antibiotic or modified antibiotics, so to say. And now for the audience, tell us what Immagina does and what are the kind of services it offers.
Yeah, the company changed quite a bit since the beginning, right? So I call our model a spinning top model. You know the spinning top?
Yes.
That's the other round. So the body center is what gives stability to the spinning top are our clients. So with our clients, there is no business model. Our clients are academic labs, so people that do research, or can be a research unit into industry. But the industry sector is then divided in medium biotech pharma and the big pharma. So these are the three categories, right? Academia, biotech, and
Exactly.
so these are our clients. The disc of the spinning top is actually our platform. So larger is the disc, more stability has the spinning top. If you have a small disc, the spinning top will follow immediately, right? And the platform is quite unique because I can say we are the only company that build up a platform based on multiple complementary, completely new technology. So it's not something that was there before and we changed it a little bit, we put it on the market, but we'll build up from scratch, meaning from science and internally, but also leveraging on the collaboration with US and European academic partner. So we build up this network of technology to study the beauty of translation and ribosome from different angles. And these are fully complementary, meaning that you may need the small antibiotic to fish out ribosomes, but you may need also a method to sequence the tRNA that are bound to those ribosomes.
Absolutely.
And that you may need a method to map the fragments that are generated by those ribosomes in tRNA fragments or ribosome RNA fragments inside the cells, or also releasing to the biological fluids, that you may need a way to use those ribosomes in an in vitro translation system to produce a protein of interest. So all this, I mean, I will not mention all the technology here, but they are all connected, and we give to our clients the possibility to buy small products so they can use the technology in their own lab, full freedom, or send us the samples and we will do the experiment for them. Or is a more like a partnership co-developments for a big milestone together. These are basically the way how we work. And we have now engaged with the most 10 big biotech and pharma company in the world with projects. So they see the value in Immagina. And so the spinning top is not finished. So we have the disc, the center, the disc. Then you have the top of the spinning model gives the force, and this is the team, right?
In his rotating, or it's the imaginary
French, otherwise nothing spin, right?
That's the force, yes.
The force, and then the table where the spinning top is playing is the market.
Wow. No, that's a very, very good analogy, Max. Just to reiterate these services or products, as I understood, so you basically enable everything around the RNA world. So you provide them the products if somebody is interested in doing the isolations or studies themselves. On top of it, if somebody wants to get these services done around this RNA space, you are also providing the services like lab services. And then you're also doing co-development, like projects where, like you say, somebody from big pharma and your team is involved and they kind of try to reach the similar milestone in a, pre-agreed way.
Yeah.
Correct?
Yeah. For example, I can give you an example that is very recent. So with a fantastic lab in Spain, we co-developed a method to fish out ribosomes. So you have your cell or your tissue, right? You fish out ribosomes, and then from those ribosomes, you can do two different analyses in parallel. One is to map which transcripts, which mRNA those ribosomes are translating, right? To infer which protein are produced, or also to understand how the dynamic of ribosomes on those transcripts. And from the same time to sequence the tRNA that the ribosomes are using during translation to have a perfect match codon versus anticodon, to understand if ribosomes are using a subset of tRNA for what they need. And this gives tons of information if you want to understand what happened in your biological model after a drug treatment, during the disease, during the progression of the disease. And for that setup, we offer the products. So the products that combine everything so that people can do it in their own lab. For example, people that have a dangerous virus, right? They cannot ship the samples to us. They really want the opportunity, the possibility to do it in their own lab. Right?
Like in-house.
And this is great. This is great because it gives complete freedom. Or maybe a small biotech company just backed by VC in US, they want to keep the project quite secret because it's still new. They want proprietary proprietary. They want to do everything in house. That's the perfect way for them to gain knowledge with a completely new solution and completely freedom. On the other side, for people that have little bit to spending capacity, but they don't have maybe the time, the resources to do it in-house, they can just send the samples to us and we will do experiment. Of course, the services, I would say, is basically really shaped on the needs of the clients. It's not like a standard CRO-like services.
So it's quite customized as per the need.
It's very customized. And this is due to the fact that everything that we offer is very new. So we have a discussion with the clients and maybe setting up a little bit different condition because they may gain more information in doing that versus that. So yeah, I would say it's very interactive, and this is very satisfying, not only for the clients, but also for the team that is actually behind all this project, working for the clients, that is contributing to success story of the client.
And Max, as I was researching about Immagina, so these are like, of course, you have the products and services, and on top of that, you also kind of offer bioinformatics solution. So, suppose a lab which is kind of doing the experiments themselves and they want to take help of your bioinformatics solution. What is on offer? Like, what do you offer as a solution? And what is the value of it, if you can just reiterate?
This, I would say, is a very big things. If you go around every CRO company, now they are offering bioinformatics solutions, support, data analysis, right? What our value proposition here is that the technology is so new, the technology that we are proposing, that also the data analysis is quite complicated and needs quite a lot of knowledge on the background. So it's not like RNA-seq. Everyone can analyze RNA-seq data using custom pipelines that are out there, right? So we are trying to support our clients in either allowing them to guiding them through what we already publish. So if they have a bioinformatician, they can also do it. But for people that want to really go fast and without having trouble with the interpretation, we have a bioinformatics unit that give full support in applying the standard pipeline, meaning getting very quick information about the QC and some basic information of ribosome function, translation efficiency analysis, which kind of protein may be produced or not, which type of tRNA, which type of RNA modification you have on your ribosome, your tRNA. But also we are expanding a lot our internal database with a lot of R&D, on which we are building, machine learning, and we are supporting also clients with an internal background of data to make their data more robust.
So basically, it's a platform that you are creating for the whole industry, so to say, right?
Yeah, exactly. The bioinformatics can be, I would say nowadays in Immagina, the BioIT unit could be a completely independent company for the value that is creating. But on together, I would say that we are quite uniquely positioned worldwide so far.
Fantastic. And coming back again to wearing two hats, right from the beginning, you are kind of a scientist and also wearing like an economic hat. And here you are like a scientist and a CEO. So how's life? Like, are you also involved in R&D projects, or you're more like driving the business, seeing the company? How does your day look like?
In the company is not like 100 people. I would say we are 20. I'm supervising the R&D project, but I have very good people in the team. So, and I would say they are quite independent. I'm supervising in a way that every week I have a couple of hours of, wrap up. I give indication. I may give strategic directional tips. I spending about 20% of my time in business development. So opening new markets, understanding from key clients and key accounts what is the need. But and then basically I'm curating very few important accounts. But if an academic lab wants to, get in touch with me because they feel more confident they can do it. They usually, a lot of people write directly to me. I may, start the first round of discussions, set up the service a little bit, or give an indication of which product they want to buy. But then there is a sales team, and that is taking tasks. They take care also because they have much more time than me in sending email and replying fast.
All right, Max. So, you have given a fantastic overview of the products and services from Immagina and about your day-to-day life at Immagina. But, like you have come a far way, like from one-person company to now 20 people company. So, of course, and it's still further growing. So, what are the current challenges that you face in the company, in running the company? Because, managing a team, managing clients, and then also trying for growth, this can be challenging, right? So,
I mean, I would say is a typical scale up issue. When you need to scale up, you would like to scale up quite fast, but there are financial constraints that you need to take into account. That are mainly related with cash flow, technology developments, so that you may want to do a lot, but then you need to select which project are more strategic. And also you have the internal organization of the team. We changed a lot in the last 3 years. We passed from a very horizontal model into a model that we shape by ourselves based on our needs. So we have a model divided in multiple assets, from research to market. These assets are basically research, development services, bioinformatics, supply chain, production, marketing, sales, and all these assets are made by a team of 2 to 4 people, coordinated by 3 coordinators that are coordinator of areas of different assets, and project managed by 2 project managers. So we have basically 3 stickers that are vertical and 2 that are horizontal. We created these metrics that create stability for the company.
Fantastic. Another challenge that I was, wanting to listen, but perhaps, you are not in that spot. So many companies, especially, companies that provide services in the biology space, including like Biotech and others, they faced basically a big challenge after the boom of COVID, right? So there was a huge growth in many companies during COVID, and then also post-COVID, you could see that boom. But then after that, the global markets have really slowed down. Are you feeling the heat in your company?
No, likely no. The reason is that this is probably thanks to our healthy balance between product and services. So to scale up product is not so complicated. So we have a clean room that can accommodate production for the next 5 years. We can scale up to more than 20 times what we are doing right now. And you don't need so many people for the production, because partially is automated, partially the way how we produce our products, that is, we do in parallel a lot of different things. So we parallelize and optimize the production in many different way. The service, again, probably this is better to get engaged with big pharma, for example, but to scale up services, you need a lot of human capital. There is no way. You can automatize some part, right? But you need still a lot of human capital every project is different. Every project needs maybe different skills. So if you are a CRO and during COVID, you get tons of requests, right? You hire 100 people, and then after COVID, you collapse, but you still have to pay 100 people, right?
Exactly.
So I think that Immagina, we basically were very healthy, and I was very strategic planning, thanks also to my board that gave us a lot of suggestion and tips, but also the structure of the company and our business model. That sounds quite a lot.
So moving to the next section, so about the future outlook and the big opportunities, so the ribosome-based technologies are actually on rise, especially with the applications in medicine and industry. You see a lot of things happening. So, what are the biggest opportunities and how do you see the future outlook?
The root is basically what is on the wall there, right? So it's a specialized ribosome. So you can see like a scheme of ribosome with different colors. That means the diversity of ribosomes. The smallest unit, by the way, is red, the largest green. And this tells you that out of the many millions of ribosomes that you have in every cells, there is also a lot of diversity.
Absolutely.
And so you are measuring something that is very abundant in every cell of your body, and very diverse. And this is basically the perfect solution for personalized medicine. You want to measure something that is very diverse, very abundant, so you can measure it every time, whatever tissue, whatever sample you have. But it gives you also a range of diversity where you can have a lot of different information from the same analysis. And this is where we are going. So our main strategic, I would say the most strategic application area are RNA liquid biopsies. So measuring those diversity into biological fluids, and this is a completely new layer for cell-free nucleic acid detection, where we can build up a biomarker made on structure, post-transcriptional modification, and sequence, and tracking back them to something that is happening on protein synthesis of the disease or the healthy, and biological drug development, new antibody, new targets, new protein targets. And of course, on the back, we have our business as usual that is supporting the RNA company to make better RNA drugs. Can be self-amplifying RNA, can be circular RNA, can be tRNA, can be standard messenger RNA, whatever RNA.
No, fantastic. So one word that is, everywhere is AI. So how are you leveraging AI? Or is there a plan that you kind of, for your bioinformatics solution, and because you generate a lot of data?
Yeah, this is quite obvious answer, because nobody will tell you I'm not using AI today, right? Everyone is using. So we are part now of this large European holding we are using AI to streamline internal processes. So this is something that we are already doing, because it's coming from this is large European holding called Technical, that is now the owner of the majority of the share in Immagina. We are actually developing our own machine learning pipeline for biomarker discovery. This is actually the big things. So all this diversity that you see here can generate tons of different features.
Exactly.
And we want to leverage on this diversity to build up the best predictive model for liquid biopsy precision medicine.
Great. So, coming to the lighter side, I think we discussed enough of science. So, we have this rapid fire round. So I'll ask you a few rapid fire questions and you have to answer without thinking. And after this section, because it's biology, career insights, you also have to give career advice, but that will be next. But let us jump into this rapid fire round. So are you ready?
Ready.
Ready. Okay. So what's the book that inspired you the most in your career?
Siddhartha, Herman S.
Wow. Padua, Bonn, Oxford or Trento, which place shaped you the most?
Oxford.
One biotech innovation outside ribosomes you're most excited about right now?
Benchtop proteomics.
Wow. Coffee or tea during long research nights?
I was used to drink coffee, now I'm drinking tea.
You are Italian, so no wonder, but then you switch to tea. Okay, that's some news. If you weren't building Immagina, what would you be doing?
I never had plan B.
So no plan B. No, this is fantastic. All right. In one word, how would you describe the future of biotechnology?
Combination now of AI and biology will generate tons of new discovery and generate values that we never imagined before. Probably biology is one of the few fields where AI can grow exponentially, because there's so much that is completely unknown that I would say, is the perfect case study for exploiting maximum potential of AI. I would say AI combined to biotech.
Fantastic. And this is the last. If you had to summarize Immagina Biotechnology's mission in one sentence, what would it be?
Oh dear, I'm not prepared for that.
Don't worry. Just summarize. That's why these questions are very spontaneous. The mission. Is the mission curing diseases? Is the mission exploring, ribosomes? What is the mission that drives you?
The mission for me is to support precision medicine with a completely new set of information coming from the ribosomes.
Fantastic. That makes sense.
So being basically the pivot and precision medicine for within the next 5 years.
No, this is a great conversation, so now coming to the final part of the conversation, there are a lot of students, biotech professionals, mid-career professionals, researchers watching this podcast. And they're all kind of, looking at one time, they, want to be a founder or they want to kind of pivot to the next level of their careers. What would be your advice to them based on your learnings?
Be brave and do whatever you like the most, and be happy at the end of the day.
Fantastic. So be brave, be authentic, and do whatever actually motivates you, whatever you like. No, that's fantastic. That's fantastic. All right. So
I didn't say anything special.
No, but Max, I mean, coming from you, so sometimes very normal things are very special because people are looking for some big goals in life. Whereas just simple anecdotes like, doing something that you like is very powerful. No, that
is the key at the end, right?
It's the key at the end. Absolutely. Yeah. Just for my own curiosity, tell to the audience about your, work-life balance, because you have a family, you have kids, you are still very young. So tell us more about your hobbies. How do you kind of keep your work-life balance?
Well, I mean, of course, it's quite obvious, but I'm working quite a lot. And,
yes, I see that.
I'm trying to, I mean, I think that is, I don't have two lives. I don't have, personal life, work life. It's one life. And I'm always working. I'm always thinking about work. I learned in the last 10 years to take some time, physically not to work, mentally I may be still working, right? To spare with the family and to listen, especially the kids, right? To listen to, because I remember at the really beginning I was so stressed. I was really running over also the needs of family, and this is not good in the long term. So my suggestion is to always keep a dedicated time to listen to your related parents, and if you have families,
loved ones. Yeah.
And also I think that it's important to also take time to work out and reduce the stress. I'm used to run, so I do a lot of trail run, standard jogging. So I use it because this is a kind of sport that you can combine in your working hours whenever you want. I go skiing, so sometimes I go skiing. For the first time in the last 10 years, I took 3 hours of during my working hours to go alone and skiing by alone, just me.
All alone.
And this was fantastic. Usually you wait for, and you take care of the kids, or you are teaching someone that just started, or you go with the company, so you are groups, right? But in the last 10 years, I never took time for just go by myself.
That's so important. Yeah.
For a good product in Q1.
Wow. No, fantastic. I'm glad we are ending up on a great note. So, thank you so much, Max, for your time. It was a pleasure speaking and learning more about you and Immagina and how you could build this company. And I wish you all the best. And we will monitor the growth of Immagina very closely, and wishing you all the success. Thank you so much.
Thank you. Thank you for your time. And it's quite late, so thanks for taking your time. You are welcome to Immagina anytime that you want to step over Austria and come to Italy.
Absolutely.
Glass of white wine for you here in the fridge.
Absolutely. And I would like to do this in person someday, that would be great.
No.
That would be great. No, this is amazing. Also, because maybe a lot of people, may want to reach you, after seeing this. So what would be your best way to connect to you? Like, is email the best way, or LinkedIn, right?
LinkedIn is probably the best way.
Awesome. So
mail get lost after the first 10. And but the LinkedIn is quite good because it can catch up maybe a little bit later. But
all right. So
great.
Awesome. So I'll put the LinkedIn, profile URL in the description. So to the audience, thank you for watching, and please follow, like, share, and subscribe Biology Career Insights. Thank you so much, Max. Bye.